Yeah, thanks, Vince.
Thanks.
Good morning, everyone. So I'm going to first start by giving a little discussion about this intersection between epidemiology and developmental neuroscience. And here now we bring in genetics as well. And I'm talking about really this area here of my work. So what is a population-based study? We have one in Rotterdam. And really, it's similar to many studies, except this was a prospective cohort design. So in inception through epidemiologists. And the inclusion criteria was mothers who were pregnant and who lived within this area of Rotterdam. That was the only inclusion criteria. They were pregnant within a certain time period and lived within that zip code. And about 10,000 mothers, pregnant mothers, were recruited into the study, and they've been followed, and now they're about 13 to 14 years of age. And it covers more than just the brain. It covers dental and ophthalmology and immunology and many different things. Also, from the standpoint of genetics, Rotterdam is ethnically diverse. And about 58% are Dutch or other European in Rotterdam in the Netherlands. From conception to currently 13 years of age, multiple measures have been collected, including blood at multiple time points, questionnaires, child behavior checklist being one of them, cognition, behavior psychiatric interviews, and MRI. And the MRI began in 2009, and it's not advancing. What's that? Okay. Yes. Okay. So wave one began in 2009, and we have 1,070 6 to 9-year-old children. Yeah. Wave two began in 2013, and we had 4,125 9 to 11-year-old children, and we're currently in wave three with over 2,000 children to date who are being scanned. I'm pushing the right button. So within the imaging genetics, I'm going to be talking about polygenic risk scores, and I'm going to talk first about behavior. So the thought of many psychiatric disorders is that they're a result of multiple genetic factors, so not one single gene, but multiple genes, each contributing a small effect. So one of the ways to look at that is by looking at, by creating a polygenic risk score, or polygenic score, because we're also going to be looking at educational attainment, and I think educational attainment isn't a risk, it's the opposite, it's probably resilience. But a polygenic score from the multiple genes, and this is all done in an independent sample from the Psychiatric Genomics Consortium. So you use an independent sample, you generate, using that data, the weighting for each SNP, and you use that for your own genetic data to create, for every individual, a polygenic risk score, for example, schizophrenia, bipolar, autism, and attention deficit. So our first study that I'm going to talk about covers behavior. So we have almost 2,000 3-year-olds and about 2,000 6-year-olds, or I'm sorry, 4,600 3-year-olds, 6,200 6-year-olds, and about 4,700. And so here's the question is, does behavior, and we're focusing on internalizing and externalizing behavior, is that related to a polygenic risk of schizophrenia. So this is before or other psychiatric disorders, but for schizophrenia, bipolar, that's before the onset of any symptoms would be expected. So let me walk you through this slide here, because so the top is schizophrenia, this is ADHD, bipolar, major depressive, ASD, and educational attainment. And this is explained variance for externalizing on this side and internalizing on this side. This is the 3-year-old, 6-year-old, and 9-year-old data. Why are there multiple bars? Because we use multiple thresholds for the p-value for including the number of SNPs that are included to create the polygenic risk score. And what we found was that even at 3 years of age, both externalizing and internalizing behavior symptoms were related to a polygenic risk score of schizophrenia. When we look at 9 to 11-year-old children, then for schizophrenia, it's really only the internalizing score that remains significant. And we find educational attainment also is related in the opposite direction. So in other words, less problematic behavior was associated with greater polygenic risk, greater polygenic, not risk, but polygenic score was related to less behavior for educational attainment. Okay, so the direction I don't have on here. So then we ask the question, okay, this is behavior. We would expect to find differences in the brain. We look both structurally and using diffusion tensor imaging, and actually the results are quite similar, but I'm going to show the DTI. We had DTI performed on 3,786, and 1,920 had genotype available. So we're down to 1,139 with European ancestry, and we're using a 35-direction DTI sequence. And here we're looking at specific white matter tracts and using probabilistic tractography and looking at the mean FA and MD within these regions. And to make a long story short, we find very large differences related to polygenic score for educational attainment with greater FA and higher polygenic risk or polygenic score for educational attainment. But in schizophrenia, and this was really puzzling, where we showed some of the largest effects, we found nothing really in the brain. And that was true for both structural and DTI. So this was a little bit puzzling. We expected to find significant differences there, and we didn't. We also found differences related to ADHD, both in behavior and in the polygenic risk. So let me move on and discuss another way that we're using polygenic risk scores, and that's to better understand different processes during brain development. So there was a polygenic risk score created for head growth or brain growth, and we wanted to ask the question, if we look at different factors during prenatal life, so between birth and between conception and approximately 6 weeks, cells divide along the ventricular zone in a symmetric fashion. And then at around 6 weeks, that changes, and you get one cell differentiating, becoming a neuronal cell, and it migrates in an inside-out fashion to form the cortical rim. And after that occurs between 6 weeks and 24 weeks, animal studies have shown if you irradiate the brain during this period, the brain will show decreased cortical thickness, but no decrease in total volume, whereas the opposite occurs from during essentially the third trimester of pregnancy, when you have a tremendous amount of growth of the brain, but also the gyrification occurs. And if you irradiate during this period of time, the cortical thickness is less changed compared to the overall volume of the brain that is done in animals. So the question that we ask is, if we have a polygenic risk score for head growth, does it have a relationship with cortical thickness? How different are those two from a standpoint of genetics? And currently we don't have a polygenic risk score for cortical thickness, although maybe now we do. I heard that there was one in the works. So what we found was actually quite interesting. This is cortical thickness. This is a polygenic score for brain size with cortical surface-based metrics. So we found what you would expect with surface area. These areas, these are different P thresholds here. And this is what we see with height, so a general growth measure. So split this in half. The measures here are with height. The measures are with or a polygenic risk score for height here. And this is a polygenic risk score for brain size. So we find strong relationships with surface area, but very little relationship between the polygenic risk score and cortical thickness until you start using many, many more genes into your model. So this is one way to use polygenic risk scores to try and tease apart different genetic factors associated with brain development. Let me finish by moving to autism spectrum disorders. So if we take the thought that autism, this could also be applied to schizophrenia, in many cases, or it has multiple genetic factors, but what about the environmental factors? So we have a large population-based studies with multiple environmental factors that have been collected. So is it also true that there's multiple environmental factors involved in autism that each contribute to the emergence of autism? And this is the environmental factors that we're looking with respect to autism were all collected during prenatal life or at the time of birth, so before a child would ever have any symptoms of autism. And I'm going to go right to the results. Here's our hypothesis, is if you have a polygenic risk score on one axis, or polyenvironmental risk score on one axis, and a polygenetic risk score on the other axis, what you would see is a gradient. So higher polyenvironmental risk with a lower polygenic risk is less risk for autism, and vice versa, whereas these would be the most at risk. So what do we find? So, I don't know, I hope you can see it, but the red dots here are the children with a diagnosis of autism in our study, and the blue dots are all the controls. So there's really quite a bit of overlap, although if you look closely, there is no one with autism with a low polygenic risk or a low polyenvironmental risk. So there might be some signal in there, and we're currently delving into some machine learning tools to look at that closer. So in summary, these merging fields of epidemiology and developmental neuroscience, they're helping to identify both genetic factors and environmental factors associated with emerging psychopathology. And so we found a relationship with very early behavior with a polygenic risk score, relationship with polygenic risk score for schizophrenia, and educational attainment, and ADHD, but we didn't find any brain differences associated with the schizophrenia, which was puzzling, but we did for educational attainment and ADHD. Although educational attainment is highly correlated with IQ in our sample, and we have shown relationships between white matter tracts and IQ. And autism spectrum disorder may be associated with multiple environmental exposures. We're exploring that more closely at this time. And there are numerous challenges I didn't talk about for large-scale longitudinal neuroimaging studies in pediatric populations. And I have 5 seconds remaining.